Good morning and welcome everybody to today's International Plant Breeding Seminar. I had the honor of introducing Professor Motoyuki Ashikari from Nagoya University. And Professor Ashikari is going to speak to us at midnight Nagoya time. So it's a first for our International Breeding Seminar. He's going to speak to us about rice breeding for Africa by using useful agricultural trait genes. He's a professor and a rice researcher at Nagoya University since 2007. He has other roles at Nagoya University, a postdoc role from 1999 to 2000, assistant professor until 2004, an associate professor until 2007. He graduated from Kyushu University in Japan. He was born in Oita Prefecture, which is part of the Kyushu Island, which is one of the most southern islands of Japan. Thank you so much for the attendance to this seminar. Thank you so much, for Professor Ashikari. We look forward to your presentation. You can start sharing your screen now.
Do you hear me?
Yes, I can hear you and we can see your screen, okay?
Okay. I am Motoyuki Ashikari in Nagoya University in Japan. Today, I'd like to talk about rice breeding for Africa by using useful agricultural genes. So cereals are main energy source for humans. Rice, wheat, maize, three major crops provides about 42% calorie human consumed. Among them, rice provides about 21% calories. I'm a rice researcher, so today I want to talk about rice breedings. So humans eat two rice species, Oryza sativa, Asian rice, and Oryza glabellimus, African rice. So Oryza sativa domesticated from Oryza luipogon, wild rice species in Asia 8,000 years ago. In Oryza glabellima, African rice domesticated from Oryza barsii is African rice in 3,500 years ago. Okay, what is the domestication? I can say it as an early crude type breeding based on the selections. So humans have selected for natural and spontaneous mutation in rice that are most beneficial to us. Today, I introduced a few traits that eliminated by the rice domestication. Firstly, traits of shattering. The seed of wild rice species easily drop down during their maturations. This trait is inconvenient for harvest. So humans selected a non-shattering rice plant. It is convenient for harvesting. Other character is a pericarp of the seed. So wild rice species pericarp of the seed color is dark brown. However, this character is eliminated through the domestication. So cultivated rice pericarp color is white. This white color looks tasty. And the other trait is their own. So this own is also eliminated through their domestication. So own is a needle-like structure at the tip of the seed. So the role of the own in wild species is very interesting. So this is a magnification of the own tips. You can see the small and fine spike on the surface of the own. These small and fine spikes enable to stick their animal furs. So own seed can follow the animal migration to enlarge habitats. Other important traits role is prevent feeding damage from predators. Okay, so why did humans select the ownless phenotype? Okay, so the own seed is inconvenient for harvesting and sowing seed. And the other important points is stocking. Okay, on seed needs larger space for stocking. However, onless seed needs smaller space for stocking. So human eliminated this character on. So far, genes regulating these characters has been isolated now. Due to the time limitation, I don't speak about the detail of the each genes. However, after domestication, human modified and improved lice by breeding, including the crossing and mutagenesis. So many countries produce own lice varieties. However, I want to emphasize a contribution of International Lice Research Institute, IRI, is located at the Philippines. So International Lice Research Institute was founded by the in 1960 by supported by the Rockefeller Foundation and the Ford Foundation and the Philippine government. So this the slogan of the ED is life science for a better world. So now at least over the 100,000 accession lives are maintained at the gene bank of ED. So that is a natural treasures. And interesting story of the bleeding in ED is a production of semi-dwarf variety IL-8. Later, I talk about this IL-8. Okay, rice is a major food for Asian peoples. Rice production cultivation system fit to the monsoon climate in Asia. At least over the 90% of rice production in the world is produced in the Asia, and most of consumption is in Asia. Rice is not only food, but it also infiltrates the Asian cultures. I believe the bleeding brings happiness to peoples. So this slide shows the conventional rice bleeding. Usually we close two varieties and produces progeny pools. And our breeders selected the better one and the ideal plants. However, late 1980s, new idea was applied to rice bleeding. So Dr. Susan McCaughey and Dr. Steve Tanksley at the Cornell University, United States, firstly applied molecular markers in the rice bleeding. So this is a paper published in the 1988, so molecular mapping of rice chromosome. Right panel show the linkage map anchored by the molecular marker RFFPs. So in introduction, they said, so the linkage map provides new opportunities for application in the genetics and the bleeding. So I think they are pioneer of the rice genomics. So this is a dawn of rice genome era. Following these achievements, Japanese Ministry of Agriculture launched Rice Genome Program (RGP) in 1991 to 1997. In this RGP program, so the high resolution linkage map was constructed, and the back library and the YAC library was prepared, and many cDNA was sequenced. After that, the Ministry of Agriculture in Japan launched the International Rice Genome Sequence Project, ILGSP 1998. This international project was 10 countries was participated: Japan, USA, France, and I can see China, Taiwan, UK, Korea, Thailand, India, Brazil. And this is a memorial picture of in 1998. So here is Professor Sasaki. He is a leader of international these programs. I was here. That time I was a PhD student. I learned many dynamics from this ILGSP project. So rice has 12 chromosomes, and color indicates the country which charges the chromosome sequence. Red color indicates Japan's chromosome Japan charged. So Japan sequence chromosome 1, 2, 6, 7, and 9, totally 50% of sequence was done by Japanese group. United States group sequence of the chromosome 3, 10, 11. And finally, 2004, rice genome sequence was completely done. I think this completion of large genome sequence is a milestone in the science histories. Okay, large genome sequence were publicly opened step by step, and the large genome infrastructure for large research also set up. So progress over the basic research was expected. For example, identification of agricultural trait genes. Firstly, here today, I want to introduce the gene cloning reveals the secret behind the green revolutions. So semi-dwarf rice variety IL-8 was born in Italy in 1960s. So this figure is show that the wild rice plants and the IL-8 semi-dwarf rice varieties IL-8. IL-8 was come from the crossing between Dijuorugen semi-dwarf variety. It is originally from the Taiwan and close with Peta. Peta is Indonesian good taste varieties. So scientists selected this semi-dwarf variety from the progeny of this process. IL-8 showed a high yielding with fertilizer, so it was propagated to many Asian countries. In totally, rice production was increased dramatically according to the propagation IL-8 with fertilizers. And this phenomena we call rice green revolutions. The semi-dwarf character is one of the most important agricultural traits because if you want to get more seed, you apply the chemical fertilizer. However, this chemical fertilizer enhances the stem elongation and the leaf elongation, so easily fall down by wind and rains. In the case of semi-dwarf variety, chemical fertilizer does not influence so much to stem elongation, influences to the grain productions. So semi-dwarf gains lodging resistance. This picture was taken in the morning of the typhoon days. So Japanese leading variety, Koshikari, was easily fall down by wind and rains. However, semi-dwarf variety could keep standing by the rain and wind and in the typhoon days. So you can understand how important semi-dwarf character is. Okay, what causes semi-dwarf phenotype in IL-8? So mutation of semi-dwarf 1 gene SD1 was selected in and used for bleeding. So SD1 firstly used for IL-8 in 1960s. However, over the 40 years, we don't know what is SD1. Okay, so our group tried to identify the SD1 genes. SD1 gene was located on the chromosome 1. Finally, we identified SD1 genes that SD1 encodes gibberellin biosynthetic gene GA20 oxide 2. So gibberellins are plant hormones regulate the stem growth. This is a structure of the bioactive gibberellin GA1 and GA4. So this is a GA deficient mutant in Arabidopsis. When you apply the gibberellin to these GA deficient mutants, the plant becomes taller like such a wild type. So you understand how important gibberellin is for the regulation of stem growth. Okay, SD1 encodes the gibberellin biosynthetic gene GA20 oxidase. This is a gibberellin synthetic pathway. So active gibberellin 1 and gibberellin 4 is catalyzed from the geranyl diphosphate GDP through the sequential reaction by the 7 enzymes. GA20 oxide 2 catalyzes a 3-step from GA12 to GA9 or GA53 to GA20s. Rice has 4 GA20 oxidase 1, 2, 3, 4. SD1 encodes the GA20 oxide 2. This is the structure of the GA20 oxide 2 genes. This is a normal type. So IL-8 has a loss of function GA20 oxide 2 type. So mutation in the exon 1 and exon 2 and leading to stop codons. So IL-8 has a loss of function of GA20 oxide 2. However, other 3 types of GA20 oxidase 1, 3, 4 is surviving. So plant becomes shorter, but not so much. Okay, this is a very interesting story. So Dijiujian is come from the Wujian in Taiwan. Okay, this is a color 76 semi-dwarf variety produced in United States. So mutagenated by gamma ray in colors. Okay, Leimei is also semi-dwarf variety come from the Fujimilari induced by the gamma ray mutagenesis produced in Japan. Jikoku is also semi-dwarf variety in Japan. Okay, so these variety are all semi-dwarf varieties and produced in the different time in the different countries by different breeders. Okay, we sequence all of semi-dwarf varieties. This is a normalized GNA20 oxide genes. ILH DGOgen has mutation 303 base pair deletion in the exon 1, exon 2, leading to premature stop codon in the exon 2. So Carol 76 has a mutation in the exon 2. So Lebe also has a mutation in the exon 3. Jikoku has a mutation of exon 1. Surprisingly, all semi-dwarf variety produces the different country in the different time by the different leaders. They have met each other. However, so all mutation occurred at the GA20 oxide too. This is very interesting. And with collaboration with Chinese group, we sequenced about this semi-dwarf variety in China. 38 out of 53 like semi-dwarf variety has a mutation at the GA20 oxide 2 genes. We found 7 different alleles. This means how important mutation in the GA20 oxide 2 is for rice bleeding. Nowadays, IL-64 and IL-72 is a mega variety is planted all over the world. This line also possesses the SD1 mutation. Now, we still using about the mutation of the GA20-oxy2. Okay, so next, I want to introduce about the other example, cloning of ill trait genes. Late plant is Nippon Ballet Japonica rice. Light plant is ST12 Tropical Japonica. This is a picture of each panicles. We count this seed number per panicle. Nippon Ballet has 147 in average. In the case of ST12, ST12 has 472 grains per panicles. Another difference of the panicle structure is primary branch numbers. So Nippon Ballet has about 10.6 primary branches per panicle. On the other hand, ST12 has 28.7 branches. It's big difference. Before of the opening large genome sequence, we could not know the gene regulated this trait. However, thanks to the genome sequence, so we could know that the genes regulating these the traits. So we found the GN1A that regulates the grain numbers, and WFP regulates the primary branch numbers. As first, I introduced about the GN1A. So GN1A encodes cytokinin oxidase CKX. Cytokinin is one of the plant hormone regulated plant growth and development. So cytokinin stimulates cell division at the flower meristem, leading to number of flower numbers. Okay, this is the structure of cytokinin. Cytokinin has cyto chains. Okay, CKX cuts this cytokinin, leading to inactive form. In Nippon Ballet backgrounds, cytokinin was degraded by the CKX enzyme to the inactive form, and the accumulation in inactive form, however, reduction of the active form, so cannot stimulate cell division at the flower meristem much, leading to lower number of flowers. In case of ST12, so expression of the CKX is very low, so cannot convert from the active form to the inactive form. So as a result, accumulation of the active form of cytokinins. So leading to stimulating cell division at the flower meristem as a result, high number of flowers. This is hypothesis. So this GNYA function was also reassessed by the other Chinese groups. So Lee et al. Produces a knockout gene 1A using the genome editing strategies. So this is a structure of the gene 1A. Their group identified many GN1A mutant by the CRISPR-Cas9 system. This is a wild type, and this is 3 GN1 knockout type. Okay, this is a wild type. GN1A-2 is loss of function because of the frame shift. GN1A-3 is functional because only deletion of one amino acid. GN1A-10 is loss of function by the frame shift. Okay, this is an evaluation of the phenotype of these plants. So we focus here today, so number of flowers. Wild type GN1A-3, this functional type, the number of flowers means the number of grain is same. However, knockout of the GN1A mutants, the grain number is larger than the wild type. So this result suggests us GN1A regulates grain number. So, as I showed you before, and the other Chinese groups also produce the knockouts of the GN1A in background of J809 varieties and the GN1A knockouts in the L237 varieties. Okay, this is a result. In case of J809, wild type is here, GN1A is here. Grain number per particle in GN1A is larger than that of wild type. In case of L237, wild type is here, GN1A is here. Grain number in GN1A is larger than the wild type. This result also support GN1 downregulation or knockout, increasing the grain number in rice. Okay, next we want to talk about the WFP, regulated primary branch numbers. So WFP means it's a wealthy farmer's particle. Also, this gene is identified as IPA1 by the Chinese group. Okay, WFP encodes transcription factor OSSPL14s. This is an expression profile developmental stage in primary branching in panicles. So blue staining means this expression of the genes of WFP. In case of Nippon ballet, we couldn't see the expression of WFP genes. However, in the ST12, so high expression was observed in the developmental stage of primary branching in panicle. So WFP expression enhances branching. So many important agriculture trait gene was identified by the rice genome sequence. So next, we expected the application to social problem. So we try to rice breeding by using these genes. So this slide shows the conventional rice breeding method. So this is a good taste rice variety. This is a high yielding variety. If we want to make a new rice variety with good taste and with high yielding, so usually we cross. And the progeny is planted in the rice field. Seedling stage, we cannot know which plant good better or not. In the summer season, plant grow. Even this stage, we do not know which plant is good or not bad, because we need to count the seed number each plant or need to taste each by each. So large field, long time laborious effort and selection skill are necessary to breeding. Okay, so how do we breed rice efficiently? So one idea is transfer gene variety A to variety B. In this case, we try to transfer gene 1A from ST12 to Nippon ballet. And also we try transfer WP gene from ST12 to Nippon ballet by crossing. Okay, this is a rice flower. This is a tiny flowers. Okay, this is a cartoon of rice seed. It is a flowering time. We remove the answers, and then we prepare the variety B and pour the pollens. This is a fertilization. This is a real crossing in the greenhouse. Okay, so this indicates is an image of the back crossing. Yellow color indicates is a chromosome variety A. Blue color indicates is a chromosome of variety B. F1 possesses 50%, 50% of chromosome A and B. So F1 is backloss variety A. And we produce BC1F1. And this BC1F1 also backloss variety A. We continue backlossing. And finally, when we do the 4x backclosing, only small segment remains in the background variety A's. So, uh, we explain this by using tomato juice. We prepared tomato juice. We also prepare the same size of the waters. Okay, so we poured in the same size of the glass, half and half. So you know that this tomato juice level is 50%. So we also prepared the same size of the glass and poured it half and half. Next stage, this glass possesses 25% of the tomato juice. And we prepare the same size of the glass of the waters and pour it at half and half. So tomato juice level is 12.5. And then we continue, tomato juice level is half and half. And finally, it's just 3% of tomato juice in the waters. This is a same strategies with backclosing. So F1 plants possess 50% of the 50% of chromosome A and B, variety A and variety B. So BC1 F1 plant possess 75% of chromosome variety A and 25% chromosome is from variety B. So BC2 F1 plants possess 88% of chromosome A and 12% is chromosome B. So continue backclosing. Finally, this branch is 97% of BC1 F1 is come from the variety A. Only 3% of chromosome is come from variety B. So important is location of the genes. Now we know that the WP gene is located on chromosome 8. So we close Nippon Valley and the ST12. If one possesses 50% of chromosome, WP region is heterozygous, and we backclose Nippon Valley, and we produce some BC1F1 progenies. Okay, all the BC1F1 progeny possess 75% chromosomes come from the Nippon Valley. However, 25% of chromosome come from the ST12. However, chromosome patterns are different each plants. So this is a structure of the Nippon Valley WP and ST12 WP. So there is DNA polymorphisms. Using these polymorphisms, we can know the genotype of the WP lesion in the BC1F1 progenies. So in the case of plant 1, so no WP genes come from the ST12. Number 2 plants also do not have the WP. The number 3 plant has a possessing heterozygous of WPs. Number 4 plants and plants do not have WP gene come from ST12. So the number 3 plant is a candidate plant for continuous backclosing. So this BC1F1 backclosed by Nippon Valley in the stage of BC2F1. So we also check the WP genotype, and we selected this heterozygous plant and backclose Nippon Valley, continuously backclosing. Finally, we can get BC4F2 generation, homozygous of WP plant genotype in background of the Nippon Valley. So we can transfer the WP gene into the Nippon Valley by backcrossing with marker acid selections. So this is nearly isonic lines of WP genes. Left panel show that the Nippon Valley chromosome. Right panel is Nippon Valley with WP genes. So we check the phenotype of Nippon Valley and the Nippon Valley plus WP. This is a panicle of the Nippon Valley, and this is a Nippon Valley plus WP. You can see the difference of the grain numbers. So we also check the pyramiding lines. So this is a Nippon Valley. This is a Nippon Valley plus WP. This is a Nippon Valley plus GNYA. This is a pyramiding line. Nippon Valley backlands plus WPGNYA. So grain number is largely changed. So this is a quantitative data of the number of grain per panicle. Nippon Valley has around 150 grains per panicle. However, the pyramiding line in Nippon Valley backlands show the 270 grain number that is larger than that of Nippon Valley. So I learned from this experiment, so introduction of the gene by backcrossing with marker assist selection can change rice traits. Okay, this is a hunger map 2020 provided by the World Food Programme, WFP. So this number indicates the prevalence of undernourishment of total populations. So darker red color is the more severe food problem is. So gray color indicates the data not available. When we see the map, so we know that there are many African countries under the food programs. So now about 690 million people do not have enough food to eat. So according to the FAO, every day 25,000 people dying because of food shortage. So this 225,000 includes 14,000 children under the 5 years of the age. So hunger is a world issue. So this is a world population growth in the past and the futures. X-axis show the years, Y-axis show the population billions. So this part indicates the last population growth, and 2021 now, world population is 7.7 billions. So this is future predictions by the United Nations. There are many scenarios. However, if we take the medians in 2060s, world population will be over the 10 billions. Now, annual population growth rate is 1.4%. However, the annual crop production growth rate is only 1%. Where is the population growth happening now? So in the futures, so now, yes, now 2021. So population growth is occurring in Africa and Asia except for Japan and China. So I think rice is a potential crop that can contribute to the food problems. So now rice demand is increasing in Africa. This picture was taken in Tanzania, Kenya, Uganda, Benin. So this is rice balls. So rice seed is necessary to the husking and the millings to eat. Okay, one bowl of cooked rice provide around 240 kilocalories. This is a very attractive energy source. Okay, so we visited several countries in Africa to assess potential of the rice bleeding by using the molecular markers. So I visited several countries. This picture was taken in Kenya. So this region has around 10,000 hectares occupied rice field. And I met researchers and I visited Mali. Mali has 100,000 hectares in the one location. It's very huge rice field. So I talked with this governor and the researchers, and I realized the problems, demands, and requests for improvement of rice. So we challenged to improve rice varieties for Africans. So I launched a program, WISH. WISH means is a hoping, but we also have the additional means, Wonder Rice Initiative Food Security and Health. This project launched in 2010 by myself. And now we invite many friends, and this is a collaboration working now, but I launched it myself. So this project is production new rice variety by using basic research and distribute visualized line to Africa and Asian country freely. As you know, that is SDG is global goals, and one of the 17 goals, zero hunger, is one of the goals. Our wish project fit to this SDG. So strategy of the WISH project is same as Marcus's selection. We selected some African variety and close with SD12 and other light species, and we backclose using the molecular markers. And then, so we introduced the important agriculture trait gene into the African rice variety. We also trying to pyramids of the useful genes. For example, high-yielding genes pyramid with disease resistance genes by crossing and the Markov selection. So when we see the genotype of the progenies, we can realize this plant possessing high-yielding genes and disease resistance genes. So this is a candidate for additional continuous work. So our collaborator work about insect resistance genes, and the other researchers working about high reading genes. Other researchers works as disease resistance genes. And we collaboratively pyramiding of these genes in the background of African rice varieties. So rice bleeding by gene pyramiding. So, but I need to say here, I care about trade-off. So we expected a pyramiding of genes leading to a good phenotype, but sometimes it's a combination leading to bad traits. At this moment, we don't know which combination is become better or worse. So now we are challenged. Okay, so we collaborate with ED because ED is located, this is a warmer place. In Japan, we can close lights a year, one times a year, but in ED, we can close 2 times a year. This is a very advantage for timelines. So we collaborate with ED and JICA. JICA is the Japan International Cooperation Agency. Okay, we produce the team in Nagoya University and in IRIS. So we selected this African rice variety for this project. So, and we also use the mega variety and other aromatic rice. Rice is very important for East African countries. Okay, so we try introduce this high yielding gene into these varieties. Also, we introduce disease resistance gene into these varieties. So this is our laboratory member at the Nagoya University side. So we plant rice in the spring season in the rice field. And in summer season, we clip the sample and check the DNA. And then, so we dig up the candidate lines and bring to the greenhouse. So we close rice and then get the seed with one seed and send to the ED. So this is ED. ED is far from the just 3 hours and a half flight distance. It's easy to go because it's a time difference is only 1 hour. And this is a project member of ED side. So they grow rice in the field and check the phenotype and check the DNA, clipping the sample and the extraction of the DNA and genotyping by DNA markers. So our lab team members is a genotype typing in each candidate rice varieties, and then digging up samples and bring to the greenhouse, and we cross. And finally, we got the many wish line seed. Okay, so we tested this wish lines in the ED. This is an example of the performance of our lines. Nerica 1 is African rice varieties, and this is Nerica 1 plus WP. This plant is Nerica 1 plus GN1A. This is a performance, each performance. So Nerica 1 is here. So our wish line, WP, introduction lines, GN1 introduction lines, so grain number plant is larger than that original Nerica ones. So performance is good. So however, I want to say important point. Even the performance is good in the ED Philippines or in Japan. However, there is no guarantee these plants show that good performance in Africa. So the important is test in the real site. Okay, so we selected the Kenya as first trial because so this is a little bit old data. So production of rice is stable. However, consumption is gradually increasing steeply. So this there is a demand of rice. So Kenya imported many rice from the Asia. However, demand is now. So this is a candidate country to collaborations. And many different cultivation styles in Kenya. This is a very important point. And there are many problems, water deficiency, rice blast disease, and a cold problem because Kenya is a highland countries and also has low nutrition problems and a salt problem. This diversity and problems represent of African rice field. So this is a point we employed Kenya's. Other reason is Nagoya University developed a research facility in Mumia, Kenya with Carlo. So he is Professor Makihara, our collaborators in Nagoya University. He developed his research facility in Kenya, so Mumia side. This Carlo is Kenya Agricultural and Livestock Research Organizations. Okay, he produces facilities, okay, and develops fields and supported by the JICA. He developed a greenhouse crossing facilities, the observation system, and developed a research field, purchased many machines, and also prepare the different condition of the field. For example, drought under the different soil type, black cotton field, slope field, salinity field, low P field, and chemical fertilizer long-term testing field. So this is a condition is very ideal for test visualize. So we started collaboration with Dr. Makihara and Kenyan governments. Okay, so last 3 years, we tested it in the visualize. Capital of Kenya, Nairobi is here. Mumia is here. About it takes 1 hour drive from Nairobi to Mumia's central area. So we also employ that location with area Busia, and we also employ the candidate test field. Mutawapa is located across the area, and we took the 3 years test in the field, okay, with collaboration with African peoples. So this is original rice variety Nerica. This is Nerica variety plus gene YAs. This plant possessing WP genes in Nerica 1 backlands. This is a pyramiding lines, gene YA plus WPs. Okay, this is so these plants were planted in the field. Okay, this is Nerica 1. This is visual line possessing gene 1 or WP. This is a pyramiding line of gene 1A and WP. You can see this plant's panicle is larger than the Nerica's. Okay, so this is an evaluation of which lines in the Kenyan field. So in the under the nitrogen fertilizer condition, Nerica 1 produced 5.2 tons, but our line is production is better than the original Nerica's. Under the non-fertilizer condition, so the pyramiding line is better than the Nerica's. So our wish line looks good. Okay, and the other wish line is based on the basmati. Basmati is aromatic rice. So you can see so rice production of Kenya, 80% of rice production in Kenya is basmati. Okay, basmati is a very favorite brand in Kenya, and however weak to cold stress and rice blast disease. So firstly, we introduced the gene 1A in the basmati. So rice production is better than the original basmati. And we also put in CTB1A, that cold tolerance gene into the basmati. So in the cold treatments, so basmati rice production is very low because of the fertility rate problems. Nerica 1 produces a good performance, show good performance. So basmati plus CTV1 performance is better one than the fertility of original basmati. So now, oh, I'm sorry. Now we are pyramiding of Gen1A and the CTB1A, CTB1 genes in the basmati backgrounds. And we also putting in the blast resistant gene now. So this is a timeline of the visualization and the seed propagation and the distribution. First batch is now under the test of national performance trial. And we expected the 2020, so this line will be legislated in the Kenyan government, hopefully. However, it's now a little bit delayed due to COVID-19. And we expected the 2023 seed propagation, and until the 2025, we want to distribute this seed into the farmers. And the second batch now is running, and the third batch is also running. Okay, so in the future, we want to expand the near country of Kenya and for the other countries. Okay, so we expected this line expanded in not only Asia, but also the African countries. Okay, so many people participated in this project. I want to say thank you, collaborators. This is a member of the WISH project in Nagoya side and the ED side. And this is a final slide. Please see this. So we wish for abundant light supply around the world and for every child to have enough light to eat. Thank you for attention.
Thank you so much, Dr. Professor Ashikari. Very good presentation, and it's interesting how with simple genes you can basically impact lives. I want to bring back a quote that you expressed at the beginning of the seminar, saying that plant breeding brings happiness to people. I think this is a wonderful quote, that simple but very wonderful. For the participants, please post your questions in the chat or in the Q&A window. We really have one question for you, Professor Ashikari. In order to locate the genes of interest in the backcrossing, do you use foreground or background selection? I imagine also the question is intended to say how can you speed the recovery of the recurrent parent with just the donor gene?
I'm sorry, I couldn't catch. I'm sorry.
So in order to locate the gene of interest in the backcrossing, do you use foreground or background selection? Basically, the question is, how can you accelerate the conversion process? Okay,
so actually, the speed up is very difficult because based on the crossing. So even in the ED side, warmer place, we can cross only 2 times. Okay, the DNA technology facilitates now and can speed up. However, this breeding is based on the crossing. So I want to speed up, but there is limitations.
Okay. Again, if you post questions, there's a comment in the chat. Thanks for your science outreach, Professor Ashikari. He's Emmanuel Brefo from South Africa, born in Ghana and pursuing a master's degree in plant breeding. I find these presentations very really useful. Okay. So several comments. There's somebody proposing that you are the next Borlaug.
No. Borlaug is established and pioneer of the bleeding of the feet. Yeah, I'm just subtle small researchers.
Yeah. One thing, one comment while I was listening to you, this approach is interesting because basically you certainly respect the preferences of people for. I mean, there's a strong preferences associated with varieties of rice, and by just changing a few alleles, you basically are maintaining the quality types, isn't it? That they don't have much effect on quality, or am I wrong?
Okay, so I haven't checked the quality of rice. So because actually I cannot evaluate the quality even we eat rice. So it's very difficult because uh, quality is influenced by the environmental condition, and so favorability is different from the people by peoples. So, and I don't mind so much of the quality because the most important point is now is production. So in the futures, I want to collaborate with other researchers working about the quality rice, but now I'm focused on the production now. In the future, we I try to improve the qualities, but now it's it's not time for the attention the quality, I think.
Okay, we have a couple of questions in the chat.
Mhm.
The fields that you do research in Africa, are there uh, associated with the national programs, or are there farmers fields?
Okay, now we are using the national field by government of the Kenyas, but so now is the size of the field is limited. So now we want to try in collaboration with farmers, and we want to test not only the governmental field but also the farmers field.
Mhm. Okay. There's a question, I think you answered that at the end. It says, when will this seed be commercially available in the future?
Okay, so I think I don't care about, I don't mind about who use these wish lines. Farmers, okay, so we can share with any companies freely. So because important point is someone use wish lines in the Asian country, African country, or other countries, we don't mind about any benefit. So I hope our line used in the farmers and any company, such as a very welcome.
Okay. Another question here in the Q&A. The person posting the question, Olabisi, is interested to know which cold tolerant genes were incorporated, and is cold stress a problem for rice in the tropics?
Okay, so yes, so in our test, CTB1 is good performance. So in the Kenya, because in Kenya is highlands, about 100, 400 meters altitude in Mumias. So I don't know, CTB1 is well in other rice genetic backgrounds. So fortunately, in the Nerika 1 and the Basmati 1 backgrounds, CTV1 can work well. But as I mentioned, so sometimes it's even we introduce the good genes, but sometimes they show that's a bad phenotype. So it's case by case. So we need to test by each. So I do not have the guarantee. So CTV1 is work in the any rice variety. We don't know.
Mhm. Okay. We have time for a couple more questions or three. Um, they're asking about the countries that you conduct research aside from Kenya. Have you already started testing outside of Kenya?
Okay, I have not started yet because firstly, we want to get the confidence of our materials. So we can do collaborate with now with other country, but we cannot regulate and we cannot manage as simultaneously in multiple countries. So firstly, we want to check and evaluate and get the confidence, and then we want to expand it to the other
Mhm.
countries.
Okay. Okay. Do you know what the acceptance is? I mean, have you done any acceptance test with consumers or local farmers?
Okay, so I have not tested it yet because we do not have abundant seed yet. But
Okay.
now is under the national trial test. Maybe next year we will ask the farmers to check say, if they favor it or not. But at least I have not checked the quality, but at least abundant production is better than the original one. So I hope farmers like our wish lines.
Okay. So what is the relationship with your project with Africa Rice is a question that we have here in Q&A. It's
Okay,
from Janot in Senegal.
okay. So I have not started the collaboration with African Rice yet. So I visited once. So as I mentioned, so we need to test it at a small site first. And then, so we get a good result. I want to start with collaboration with the African Rice. But now we need to check the phenotype and evaluate our line first.
By the way, a couple of persons have requested the contact information for Professor Ashikari.
Mm.
I have posted it in the chat, but we will probably post it when we send the next reminder and with the link to the recordings. Okay, there will be his email address. Okay?
Yes, please give me the
Yes,
by
yes,
emails. I'm happy
yes.
to answer.
So finally, there's a question here about what is the budget for all this research? Is it
Okay.
huge? Is it affordable?
Okay. Honestly, the project is started in 2012 supported by JICA. That's a project of 5 years. 2018, this project was done and no supported now. So now we got this small budget from the Japanese government. So now I'm trying to get a budget from Japanese government and to push more. So we can get the budget outside countries probably possible. But as I mentioned, firstly, I want to test in a small size, and then we want to expand. So now is a small budget is okay to evaluate in the Kenyas. So in the future, we will get this huge budget to expand in the other country in Africa.
Great. Okay. There's a lot of messages. Thank you for your presentation, Professor Ashikari. And thank you so much for taking the time at this particular odd time at your place. The recordings will be available and will be made available to all the participants pretty soon. Thank you, Brandon and Anthony, for your help in setting up the links and the logistics. And next week, we're going to have the 5th seminar of the seminar series is 6 seminars. So we have 2 more to go. Dr. Susana Emilia Lewis from our university here at NC State will talk about turf grass breeding, one of the most successful emerging programs in our university. Thank you so much to everybody. Thank you so much, particularly to Dr. Professor Ashikari and Tomohisa here at NC State, another North Carolina, another representative from Nagoya University, who helped us make the connection and help bring this seminar to life. Thank you so much, and you guys have a excellent rest of the week, and I look forward to seeing you next time when we meet in a week for the next seminar. Thank you so much. Bye-bye.
Thank you so much. Bye-bye.